All right, in this video, we'll continue the second half of Unit 6, beginning by talking about genes and development. And then we'll be moving on in this second half of the unit to talk about meiosis and then Mendelian genetics, which is kind of the principles of heredity all together. So, in order to talk about this, we first need to talk about a few vocabulary terms, which is basically all we're going to do today. Just talk about vocab. And so first you have this structure called a zygote. A zygote is a diploid cell. Diploid means that it has both sets of chromosomes, or one set from the male or one set from dad, one set from mom. And so a zygote is a product of sexual reproduction in that it has two pairs or two chromosomes in each pair. It's diploid in that. And over the course of an organism's development, they will change, and that zygote, that single cell, will become many different cells, and those cells will then specialize. And this is called cell specialization or cell differentiation. And basically, what this is when a cell expresses different genes, and some genes say you're going to go be a skin cell, some say you're going to go be a blood vessel, or whatever it is. That cell grows up and then becomes a certain sort of cell. Here's an example of a specialized cell. In the nervous system, of course, very specialized, looks so much different than the other cells. And you can go back here and look at the skin cells, how they're nice and flat and plain. This cell has a particular function, and it's that specialization, the turning on of certain genes and the turning off of other, add to that. All right, so this concept of morphogenesis, again, talking about development, so considering all that in context, morphogenesis, morph means shape, genesis means like the creation of, and so this literally means the creation of a certain shape. This is going to be different depending on whether or not you're an animal or a plant. In animals, that shape is going to basically begin the same as the zygote, that fertilized egg, and that is going to just kind of go from a ball of cells. We talked about this in Unit 5, and some of those some of those structures are very similar. Remember the balastula and the gastrula, and then this is a pretty dramatic jump here, but some animals really don't get out of this stage. They kind of just become this inward folded cup, like a jellyfish, for instance. But for other animals, it becomes more complex. And then plants, obviously, very similar, still have a zygote, but then there's going to be some more differentiation there that makes them plant versus an animal. Same idea, though. This sort of shape change is dictated by the DNA. And that's what this idea of apoptosis, something that we've talked about before, has to do with programmed cell death. It determines the shape as well. So you take like a hand, for instance, used to be kind of this paddle shape there on the left. And then because of apoptosis, programmed cell death, a cell knows it's time to go because it is the skin that exists between the fingers. And it's just not supposed to be there. So that makes sense. And there it is in plants. Same sort of thing. In order to make those little root hairs or those little leaves, certain cells have to die, other cells have to grow, the shape has to change, and all of that is coded for on the DNA. And more of that. All right, so let's talk about cloning. Cloning is basically a process in which there's another organism that is made that is 100% identical to the original. This was first made popular by this sheep here, whose name was Dolly, in 1996 and 97 is when this took place. And since then, lots of animals have been cloned. And but the same idea is basically true. What they did is they took a cell from the donor. They took a mammary gland cell and basically remove the nucleus from it, because the nucleus of that mammary gland says, hey, you're a mammary cell, right? Just think from back here at this cell specialization. You're a mammary gland cell. You only know how to be a mammary gland cell. Well, they need it to be all the cells again. So they took the nucleus, or they took an egg from another cell, from another donor, and put the mammary DNA inside that egg. And so the mammary cell DNA then basically kind of reset. in a sense, and became a set of DNA that was then able to become any kind of cell. And they implanted that embryo into the surrogate mother, and lo and behold, Dolly was born. A lot of times when people think of cloning, they think of like going to the copy machine and making a copy of an animal, and you have another adult animal sitting there. No, when you make a clone, you start with a single cell, that zygote again, and it has to grow up, but identical DNA. So let's talk a little about stem cells. Stem cells, and we mentioned a little bit about that in the previous slide, just didn't call them that. A stem cell is said to be pluripotent, meaning they have the potential to become any kind of cell. Sometimes you will see the word, I think I have a picture here of this too. Yeah, sometimes you will see this called totipotent, which just, totally potent. Potent just means powerful. And so think of it like becoming any kind of sort of cell, whereas pluripotent is more along the lines of like a cell in your bone marrow can become any kind of blood cell, versus a totipotent cell can become any kind of cell. Sometimes you'll see pluripotent used both ways, but there's just an exposure to both of those words. And the idea behind stem cells, again, is that in a stem cell, the DNA is not as closed off. It can become multiple kinds of things. Whereas you take a cell, for instance, like a bone cell, since we're looking at bones here, bones only know how to become and do one sort of tasks or one set of tasks. They know how to do bone cell things. They have the DNA to do all the cell things, but they just have all that stuff turned off. And we looked at that in the previous half of this unit, how cells can turn on and off information. This same kind of thing leads to something that's called pattern formation, and this is in plants and animals, that there are certain patterns that exist on the DNA that are going to come about because of the DNA, because of those genes saying you be shaped like this versus being shaped like whatever else, different kinds of leaves and plants. Think about the way an animal is shaped. There's not a lot of difference between a mouse and an elephant. If you look at a mouse and an elephant, obvious size differences, but they have all the same basic parts and the same basic pattern. It just happens that an elephant has a little bit of tweaking and it causes this giant difference. And so there can be a lot of change and a lot of tweaking that can exist in these pattern and another set of positional genes. There we go. And the positional genes just says this is where things are. And so if you think about pattern genes, this is the shape of things. Positional genes, this is where things are located. You can tweak those things to cause a lot of different patterns and ideas in an organism. It doesn't, even inside of a single species, there can be variation. If you look at a person's face and you look at someone else's face, their eyes aren't exactly in the same location and so forth. And so you can kind of see how there can be a lot of subtleties in this information. But again, all coded for in the DNA.
Testing 1, 2, 3.
All right, so
Testing 1,
ignore the
2,
testing.
3.
All right.
Peaceful day here at my house. Sunny skies, clear. Temperature of 41 degrees.
Okay, there's that. I don't know if you heard that or not, but I did. All right, so real quick, finish up before that happens again. So this idea of the maternal effect or egg polarity, basically what's going on here is early on in development, the egg and the zygote will develop poles, and the animal and vegetal poles will become the head and the not head, respectively. We've talked a little bit about this in Unit 5, but this is just to show you that there is some differentiation that begins way early on in the zygote. And this is another picture that these cytoplasmic determinants are what cause this to occur. There are particles inside the cell, and those particles kind of go to one side or the other. And then when that cell divides, well, the cell on the left got all of the head particles, and the cell on the right got all of the not head particles. And so you can see where that differentiation can start to occur really early on in development. And this is what leads to, as you get multiple groups of cells, like the ball of cells, remember the morula and the blastula and the gastrula, those cells are going to start to determine themselves more and more. And so one whole group of cells is going to go off and become the liver. And then this other group of cells that's right next to them is going to go off and become the gallbladder. And how do they know? Well, they just know that they're not the liver. They're supposed to become the gallbladder, and they recognize where each other is. And the position of one is very important concerning the position of another. If one of them gets off, then it can actually cause this chain reaction, and all of them can be kind of broken. So it's very important for them to all be touching and talking to each other in this communication. Very important, very early on. And then lastly is this idea of Hox genes. Sometimes you'll see them called homeotic genes. Hox genes is what I've seen, HOX, most of the time. And these are kind of the master control genes of an organism. There's a switch, and think of Hox genes as switches. Maybe I have a picture. I do. Awesome. Think of a Hox gene as like this master control switch that's going to then turn on all these other switches. And this switch may say ears. I don't know. Let's see, not ears. Let's say eyes. Well, this switch is the blue eye, or the color button. This is the shape button. This is the, I don't know, something else button. I don't know. You get the idea. So this is just the master control eyes, and these other ones control lots of different stuff. Well, a lot of the variance that exists from one species to the next is found in these switches, because an elephant may have different switches than a mouse, obviously, or what really happens is they have more of these switches. An elephant just has more ear switches than a mouse does, and so that's why an elephant has more ear. This is an oversimplification by all means, but understand it's basically that simple. There's just a master control that kind of sets all of them off. And then so when you're done making ears, what's the easiest way to turn all these genes off? You just wrap up the Hox gene, and all the other things just kind of stop making ears. And so you go back to what we were talking about with stem cells all the way back here. The stem cells have the ability to flip all of those switches either on or off. And so that lends itself to lots and lots of possibility.